All right, today we're jumping into a real medical detective story. We're talking about a group of rare cancers that are just masters of disguise. I mean, you look at them under a microscope and they seem totally harmless, but they're hiding this dangerous, sometimes fatal secret. So how on earth is that possible? Well, let's find out. And this quote right here, it really gets to the heart of the whole puzzle. You've got a pathologist looking at this tumor, a low-grade fibromyxoid sarcoma, and under the microscope, the cells look, well, harmless. They call it low-grade. But then, years, sometimes decades later, it can just suddenly spread. It's this exact paradox that has had doctors scratching their heads for a very long time. So that's the big question, right? How can something that looks so benign be so incredibly dangerous? Well, it turns out the answer isn't in what we can see, it's in what we can read. We're talking about the tumor's hidden genetic code. Okay, so our first clue is actually right there in the name, myxoid sarcomas. Figuring out what myxoid even means is kind of the first step to cracking this whole thing open. So, myxoid, it sounds complicated, but it just means these tumors make a whole lot of this gel-like, almost slimy substance. It's called an extracellular matrix, and it's packed with these complex sugars. But here's the kicker. Even though they all share this gooey texture, their behaviors are completely different. I mean, some are super slow growing, and others, they're incredibly aggressive, worlds apart. So, to get to the bottom of this unpredictable behavior, scientists realized they had to move beyond just the microscope. They needed a totally new way to see what was really happening deep inside these cells. And this right here, this is the game changer. The old way was called histology, and it was all about appearance. What does the tumor look like? But the new way, genomics, is all about reading the tumor's instruction manual, its DNA. It's the difference between judging a book by its cover and actually reading the story inside.
Yeah, the big problem with only looking at appearance is that some of these different sarcomas are basically doppelgangers. They look confusingly similar. You could have a low-grade Myxofibrosarcoma that looks almost identical to a low-grade fibromyxoid sarcoma, but how they behave? Completely different. It really is like trying to tell two strangers apart when they're wearing the exact same coat. You've got to know what's going on underneath.
The real breakthrough came with the discovery of something called gene fusions. Think of it like a unique molecular fingerprint. What happens is, two totally separate genes literally break, and then they get stitched back together, but incorrectly. And this creates a brand new hybrid gene that can just drive cancer growth. This became the definitive clue, the smoking gun, to finding the tumor's real identity. So, armed with these new genetic tools, we can finally start to unmask the different members of this myxoid sarcoma family. We can see what really makes each one tick. All right, let's put two of the main culprits side by side. First up, we have Myxofibrosarcoma, or MFS. This one usually shows up in older adults, and its genome, its entire set of DNA, is just a chaotic mess of mutations. Then you have low-grade fibromyxoid sarcoma, LGFMS. This one tends to affect much younger patients, and it's not driven by chaos, but by one single very specific genetic event. Okay, let's dig into Myxofibrosarcoma first. If its genome were a crime scene, it would just be pure, unadulterated chaos. There's no single smoking gun here. It's more like a riot of genetic mistakes all happening at once. Scientists have a name for this: extreme chromosomal instability. And this chart really shows you the key players behind all that chaos. The biggest one by far is a gene called TP53. Look at that. It's altered in almost 57% of these cases. Now, TP53 is famously known as the guardian of the genome. Its job is to stop cells from growing out of control. So when it gets broken, it's like the brakes on the cell are just completely cut. And you can see other guardian genes like ATRX and RB1 are also taking a hit. This isn't just one little mistake, it's a total collapse of the whole system. Okay, now let's flip the script and look at the fusion-driven sarcomas. These are what we call genomically simple. So instead of that total chaos we just saw, here you have a single identifiable genetic event that kicks the whole thing off. It's a much, much cleaner crime scene. And this is the smoking gun I was talking about. In more than 90% of low-grade fibromyxoid sarcoma cases, what happens is the fused gene from one chromosome gets fused with the CREB3L2 gene from another. This creates that brand new hybrid gene, fuse CREB3L2, and that is the single thing driving the tumor. It's the definitive fingerprint, no question. And it turns out LGFMS isn't the only one playing this game. Myxoid liposarcoma also uses that same FUS gene, but it hooks up with a different partner, a gene called DDIT3. So it's a really similar mechanism, right? But that slightly different fingerprint, that FUS DDIT3 fusion, ends up creating a totally different kind of cancer. Okay, so this is all really fascinating science. But how does knowing about these molecular fingerprints actually help patients? Well, it completely changes the game. It revolutionizes the diagnostic toolkit and gives doctors this incredible precision for identifying these tricky tumors. It's pretty cool how it works. That FUSCREB3L2 fusion gene, it acts like a light switch, flipping on other genes that are normally supposed to be off. For instance, it just massively ramps up the production of a protein called MUC4. So what does that mean for doctors? Well, now they can just take a tumor sample and stain it for MUC4. If it lights up, boom, they have a nearly 100% certain diagnosis of LGFMS. You've got a direct line from the genetic cause to a simple, super reliable test. But, getting the right diagnosis is really only half the battle. The ultimate goal of cracking a cancer's genetic code is to find its weak spots, its vulnerabilities, and then design smarter therapies to go in and attack them. And this is exactly where the future is headed. For those chaotic sarcomas with the broken TP53 gene, there are these new drugs called MDM2 inhibitors that are designed to try and reactivate that guardian of the genome. And for the fusion-driven ones, like myxoid liposarcoma, there's this absolutely groundbreaking new therapy where they actually engineer a patient's own immune cells to hunt down and kill the cancer. You see, each therapy is precisely tailored to the specific genetic code that we've now cracked.
And all of this brings us to the really big takeaway message. For decades, we defined cancer by where it was in the body, or what it looked like under a microscope. But we're in a whole new era now. It's the era of precision medicine. And in this era, the most important thing about a tumor isn't its name or its appearance, it's its unique genetic code. It's what's driving it.
So it really leaves us with this powerful question for the future, doesn't it? We've managed to crack the code for these rare sarcomas, and we've turned a medical mystery into actual molecular targets. So as we continue to map the genome of every single type of cancer out there, you have to wonder, what other secrets are just waiting to be unlocked? What other puzzles are we about to solve?